Alright, let's continue on with two types of gene therapy that I'm going to cover, which is adenovirus and retrovirus gene therapy. So, of course, the adenoviruses and the retroviruses used has actually been recombined, which means that modification has been done to it. And for adenoviruses, the recombinant adenovirus for gene transfer has been found to be efficient. And the first recombinant vector has been engineered in 1985, and it has been used for clinical trials. So some say that, mouse a clinical trial basically is okay for mouse for gene therapy or whatnot. Yes, okay, so it has been done. So the clinical trial application was carried out in early 1990s. So let's take a look at the advantages of adenovirus-mediated gene transfer. Among them is can be produced efficiently at very high titers, which means that at very high concentration or quantity. It is capable of encapsulating DNA molecules 6% bigger than its own viral genome. It allows introduction of large DNA fragment up until 7 to 8 kilobyte, which is basically is quite big as compared to normal vector, especially plasmid vector, whereby you could only include or insert up until 2 to 3, which is also quite large. The next one, it can infect non-dividing cells, which we know the property of adenovirus. It allows high transient overexpression of transduced gene, which means that the expression is actually lasts a bit longer, and it also allows for overexpression. What it means by overexpression is that when you have a certain genes and when you have it to be overexpressed or doubly expressed, so that would be good. Certain viruses couldn't basically have this feature. The next one, it remains epichromosomal, therefore no insertional mutagenesis, which means that it remains more or less like its own state, and has been found that no mutation, no adaptation has occurred, so it doesn't change its own feature, and it can be propagated in suspension culture quite easily, and there are updated and modified and of course established method. So the possible application of adenovirus gene therapy is in, of course, transfection, whereby it can infect most cell types quickly. It doesn't really matter where the cell types comes from, because there are certain cell types, especially for human body, which is quite hard to be transfected, such as those coming from the lung. So analysis of gene expression can also be analyzed after a few hours of infection because of the long transient expression. So expression of more than one protein can also be done simultaneously, and this has been proven by some journals produced. Secondly, overexpression in human cells. So that is one of the advantages which was in the previous slide. So it allows for production of high levels of heterologous protein. The protein produced represent abundant polypeptide, which is of course useful for the protein to be produced to basically more or less perform its function. And of course, the expression system can yield up large suspension cultures, which means that there is no problem at all cultivating it in the lab. So of course, further development has been done, and this has been tested on muscular dystrophy. In case you don't know what muscular dystrophy is, if you have seen someone with a hunchback or whatever, so those actually is caused by muscular dystrophy, cancer therapy, hypercholesterolemia, and neurological and vascular disorder. So basically what happens is that from up until 26 onwards, so the strategies are basically to develop high efficiency transgene expression vectors with short-term immune suppression generating chimeric adenoviruses, which means that they change a bit of the adenoviruses but make sure that it's not being detected by the immune system. So when they did it in the animal lab, mouse adenovirus has also been administered and found to provide potential alternative to human-based adenovirus. But of course, we have seen some advantages. So let's see some of the advantages or limitations to adenovirus gene therapy. So it is basically highly immunogenic, which may cause toxic reactions in hosts, especially if you were to more or less overexpress. Expression of gene product may trigger immune response because of its high immunogenic property. It may only last for a few weeks. That's why you need to more or less repeat or go back for a backup, and difficult to remove viral genes from the vector, which is what they encounter in the lab. And of course, there is a response in terms of acute toxicity, which may occur at high doses from viral particles. So they actually have to be very careful on the doses released in the patient or given to the patient. And the last one is that most vectors this has been found to be true, ends up in the liver through IV if it is through in vivo. What happens is that the vector is not destroyed. So when it is destroyed, so basically there is a mechanism in our body that will basically take it to our kidney to be flushed out. But this vectors, if it's through IV, most of the time it's end up in the liver, and when it infects the liver, usually, well, simply say, you don't really have that long to live. So this is the case of gene therapy using adenovirus vectors. So this is actually a very common picture. You can Google it and you can find it. So remember the mechanism of gene delivery via gene therapy. So this is adenoviruses. So your transgene has been inserted into it. So what happens is that before it enters into the host, it will bind to the host surface first, because remember it has the receptor to bind to it. So later on it will make a hole, basically make sure that it remains in endocytosis. So later on once it has entered, it remains there and make sure that it is safe from being detected by the immune system. Then later on it will release itself, and of course the virus by right should release the transgene, and the virus should be degraded, disintegrated, or making its way to our kidney to be disposed of, and your transgene should make its way into the nucleus, and of course hopefully integrate into the chromosome and replicate as one. So for the retrovirus gene therapy, so let's hope you can see some of the similarities and differences. So between them, retrovirus and adenovirus, retrovirus has been found to be more efficient as compared to adenovirus, especially for mammalian cells, and the most common vector is based on MMLV. So I'm sure, hopefully, you still remember from gene therapy, MMLV is myeloid leukemia virus, and currently another type of retrovirus used for gene therapy is also HIV. Well, of course, in the hope to cure HIV itself. So interaction occurs between specific receptors on the host cells with envelope proteins of the virus, and it has been found that they actually, not to say do not, but low immune response. They do not elicit high immune response in hosts, but are of course susceptible to rate degradation, which means that they are attacked by degradation in our body. So for this kind of gene therapy, it is mainly produced by transient expression, and of course some of the advantages offered by using retroviruses are it provides high titer range, so that means that both of them have these similarities. It has rapid production and highly reproducible, so no problem there to make sure that your result is valid. It allows long-term transgene expression, usually more than 24 hours, that has been reported, and able to integrate efficiently into the chromatin of target cell, which means that it should be no problem to be integrated and replicate as one with the host. So the possible application of retrovirus gene therapy is for maintenance of genetic information in self-renewing tissue, and this has been done. Vector designed for transgene delivery under the control of tissue-specific promoters. It is quite hard, but in mice, it is quite efficient, and facilitate in vivo retrovirus-mediated gene therapy. This is, of course, in the work. So, of course, further development has been done. This is to produce new retroviral transfer vectors to maximize the transgene expression, and of course to make sure that chimeric retroviruses could also be engineered with specific cell tropism, which means that it depends on the cell's condition, environment, and stimulus. So the current application up until now are for the transfer of HSV-TK gene into donor lymphocytes to control leukemia response, and of course transduction of cytokine receptor beta chain to induce immune response. Gamma chain. So focus in on the replacement of ENV gene. So remember the ENV from the retrovirus property with another MLV to change the vector's tropism, of course extend to make it a wider host range. But of course there are certain disadvantages or limitations in terms of productive transduction of retrovirus depend actually on target cell mitosis. So it basically lacks infection in non-dividing cells, which means that retrovirus on the whole couldn't be used to be delivered into non-dividing cells, but some of its family do have these properties such as antiviruses and whatnot. So integration into host genome with random insertion might interfere with gene regulation of host cells, because it has been found that sometimes it may take over the host cells gene regulation, and dependability on cell division. which of course, like I said, okay, it works more efficiently, okay, on cells or tissues that are dividing. So it restricts transduction to limited target cells. Alright, so there you have it. Okay, go ponder on it, see some of the similarities and the differences, and then move on to the next video.